My name is Craig Jackson. I was at one time professor of biological chemistry at Washington University School of Medicine, and it was from work that I did at that time and earlier that I came to know of Peter Esnouf. I first met Peter in person in 1968, although I certainly knew of his published work because of my own graduate studies. But I contacted Peter and traveled to Oxford to introduce myself because of our common interest in factor 10. And Peter was the first individual who isolated factor 10 in a large enough quantity to actually be able to start doing physical and chemical characterization of it. Well, our friendship was extended actually when Peter invited me to a meeting of the Royal Society, which was in 1968 in London, which had been organized by Gwyn McFarlane. Title was Triggered Enzymes. Peter presented a paper there that I think should be a classic, although I think it's probably not noted. This paper actually was one of the earliest comprehensive considerations of structure-function relationships within coagulation proteins. Peter was a very straightforward and honest investigator, and included in that presentation was a discussion of the differences between prothrombin, as was prepared by Walter Seegers and his co-workers, and factor 2, using the two designations to describe a fundamental distinction. Seegers prothrombin could be converted to thrombin in the presence of 25% sodium citrate, now known to be due to the fact that Seegers prothrombin was the prothrombin complex, whereas factor 2, which had been further purified, remained intact under those conditions. That was a key observation, and one that, again, I think wasn't appreciated to the extent that it should have been. That talk, Peter also discussed the challenges to the then popular waterfall model of Ratnoff and Davy and the equivalent cascade model of Gwyn McFarlane with respect to the question of factor 5, was it an enzyme or was it a cofactor to factor 10A? Well, it was somewhat speculative. Not all the data existed that we have today, of course, and yet Peter, with his foresight, identified the potential molecular mechanisms by which factor 5 or factor 5A could be accelerating the conversion to a prothrombin to thrombin. He suggested things such as the adsorption of the components of prothrombin activation to the phospholipid surface. And in fact, phospholipids in coagulation was Peter Esnouf's DPhil dissertation. He suggested also that alterations in the conformation of factor 10A and or prothrombin would increase the rate of prothrombin conversion to thrombin. What we know now, of course, all of these turned out to be, in a sense, prophetic. It took another decade or so to actually come to understand them and appreciate them. In all fairness, however, it needs to be noted that the basic insights into prothrombinase were really presented by Haskell Millstone in 1964. And that was a seminal paper that, again, is lost to some extent. In 1967, he actually began work on the enzymes of the venom of the Malayan pit viper. For those of you who are old enough to remember, there was a product called Arvin, one of the early fibrinolytic agents that was clinically used. This was the product of Peter's work. Well, Peter's work on vitamin K related proteins continued. Some studies of vitamin K action and some papers on the heterogeneity of vitamin K dependent proteins that could be isolated from cattle that had been given vitamin K antagonists. Probably what's best remembered from Peter's lab, although not necessarily remembered for Peter's contribution, was the discovery of protein Z, or protein Z as he called it, with Chris Prowse, who was one of Peter's DPhil students at Oxford. Well, Peter sort of shifted gears a little bit in the 1980s, and with two of his friends, George Miller and Tom Mead, Peter worked on factor VII and its actual possible role in thrombosis. He developed an assay that was used in the epidemiologic studies that George and Tom were responsible for. And Peter, along with his wife, Annette Burgess, had an entrepreneurial streak, and they formed two companies based on laboratory work, which led to collaborations with Shield Diagnostics, which was in Dundee, Scotland, and Chugai Pharmaceutical Company, which was in Tokyo. So Peter was pretty broad in what he was doing. In the actual obituary, Annette and I point out that Peter had several other interests as well as science. He had a Radio Oxford show on the arts, and he was actually the head of the Oxford Theatre Company for almost 30 years. Well, Peter and I lost contact for several years when I moved from Washington University at the American Red Cross. Well, Peter and I met again a few years later at a scientific meeting, and at that meeting we renewed our friendship, which has then continued until Peter passed a couple of months ago. Well, we actually then began what came to be a tradition of having chats every week or so via telephone. We could reminisce and chat and talk about ongoing work. Due to some work that was introduced to me by Swedish investigators, Peter and I then collaborated on writing a challenge to the prothrombin time, and this resulted from data on a comparison of the quick prothrombin time, or what we just call the PT in the US, and the Oren prothrombin time. Well, later, when I moved to San Diego, which is where I now live, I became very interested in calibration of reagents in clinical chemistry because I headed an IVD manufacturing company for a few years. Well, that got me involved in something called the Joint Committee on Traceability in Laboratory Medicine. And as a result of an invitation to try to introduce traceability as seen by clinical chemists into the hemostasis testing, I invited Peter to join the group, which was working on better assays for antithrombin. Well, unfortunately, collaborations with the SSC of the ISTH and the IFCC, which had been tried one time before, were not particularly successful. There are two philosophic differences with regard to laboratory testing, which served to be an impediment. Later, Peter visited me in San Diego at my invitation when I asked him to comment on instrumentation development. And why did I ask Peter? Well, he developed instrumentation. He built his own computers in the early days, and I knew that I could get candid, honest advice from him. I have to say probably that our mutual respect was one of the bases for our more than 50-year friendship. And we both conceded retirement, that we could do what we thought was right. We were no longer subject to political considerations. Maybe wise, maybe not. Well, up until Peter passed away, we had, over a period of probably 4 or 5 years, weekly Skype sessions, video conference, which we could chat. Annette Burgess Esnouf, his wife, then I could chat. And these continued actually up until the week before Peter made his final trip to the Churchill Hospital in Oxford and his passing. What I would say really in conclusion is that Peter was a fabulous friend, and I personally really miss those calls.